So for my project this summer, I studied tomatoes, and there's two types of tomatoes here. We've got this one, which is your normal tomato that you'd find in the store, and then you've got this one, which is a wild tomato. So the thing about your normal tomato that you'd find in the store is that while it tastes pretty good, it doesn't have a lot of genetic diversity because it's been selectively bred for a really long time to taste good. So all of these tomatoes are really similar genetically, which is not great because we want more genetic diversity because it means that there's more room to improve on the flavor of the tomato. On the other hand, the wild tomatoes don't taste so good, but they do have a lot of genetic diversity. So what we did in this project was we crossed these two tomatoes, the wild tomato and the domestic tomato, and we bred them together to get a cross, a hybrid. And from this tomato, we're trying to find what genes might be differentially expressed between the allele from the domestic tomato and the allele from the wild tomato. So for each gene, we're trying to figure out whether there's more expression from this parent allele or that parent allele. And what that'll basically tell us is which genes are controlling for flavor and nutrition, because we can taste the tomato and we can taste these tomatoes and we can compare. So what we did was, in order to find the expression, we took RNA reads from this cross, and what those RNA reads basically tell us is the expression of each gene. So we can take each read for each gene and we can compare it to the genome of the domestic tomato, we can compare it to the genome of the wild tomato, and then once we do that, we can assign it to either the domestic one or the wild one, depending on how well it aligns with each one. And then for each gene, we'll have a count of how many reads were assigned to the domestic one, how many reads were assigned to the wild one. And that'll tell us the different expression between the two alleles. So if this one, say, had 100 reads assigned to it for a particular gene, and this one had 1 read assigned to it for a particular gene, then we know that this has a lot more expression. So once we split up all those reads, you can maybe kind of see over here that there's 2 big clusters over here, so they do separate out. This is a principal component analysis diagram, and you can see that this principal component along the y-axis is really just the parents. We've got 2 big clusters right here, and then this heat map also shows you something similar, which is that all the reads that are assigned to the wild one correlate quite nicely, and all the ones that are assigned to the domestic one also correlate quite nicely with the red and orange. So once we do that, we can look at the genes in particular that are differentially expressed between the alleles that relate to flavor and nutrition, because that's what we're interested in a tomato. And we can find these 24 genes that have significant differences in expression between that allele and are related to flavor and nutrition. And what we find is that, for the most part, they're really all expressed from the domestic allele. So you can see that this kind of chunk here of red and orange, that's all from the domestic allele, and then these right here is from the wild allele. So you can see that it's a lot of red and orange over here. So kind of comparing these expression differences and then comparing the taste allows us to get a better sense of what controls for flavor and nutrition in a tomato, and we can also do further research looking deeper into these genes and maybe deciding whether they're a good thing for taste or they're a bad thing for taste, whether we want more expression or less expression, and we can figure out how we can add more genetic diversity to the domestic tomato while keeping a good taste. This was kind of something that I expected before coming to a research lab, but in a way, I think being there and having everybody else working on all these totally different projects while I was working on my project was in a way kind of surprising, because I've got my own thing and then I've got all these other people who are doing totally different things that are all really fascinating. It was just really great to be exposed to all of that.